Hello to everyone. Myself Dr. Deepika Saini, Associate Professor at Lloyd Institute of Management and Technology. I am going to discuss further antipsychotic agent. In the last class, we did the common mechanism of action that is possessed by antipsychotic agents, and apart from that their classification, how these antipsychotic drugs have been classified, like we did phenothiazine category, the structure activity relationship of these drugs. Further, we will now study the different classes, the remaining classes of antipsychotic agents. So, the first of all, the phenothiazine derivatives, the promazine, chlorpromazine, we already did in the previous lecture. Now, the next category, that is fluorobutyrophenones. Fluorobutyrophenones, they are actually, they were initially they were known to be analgesic. They were they were developed during the development of analgesic derivatives. So, the fluorobutyrophenone, we have three examples here. Haloperidol, one of the most important drug that is used as antipsychotic agent, droperidol and risperidone. So, these are the drugs which are being used as antipsychotic agent as fluorobutyrophenone. Now, we would like to study about the structure activity relationship of fluorobutyrophenone. As the name suggested, we have different type of modification available, like the first one is phenone position, that modification at benzoyl group, that is phenyl ring associated with a carbonyl group. So, the first target is benzoyl group, where the modifications can be made. The second one is amino group modification, that we can modify this amino group. And the third position is modification at the linker group, this CH2 variation, that how can we vary this bridge, this linking CH2, this methyl linkers to get different activities of compounds. The first category is modification at benzoyl group. First, what is benzoyl group? As we know, when we remove a single hydrogen from benzene, it's phenyl. Generally, students get confused between these three different structures. When we add a CH2 group here, this is benzyl. Similarly, when we add a carbonyl group here, so when we add a carbonyl group here, this is benzoyl group. So, with a benzene ring, when there is a C double bond O group, that is benzoyl group. Now, at this where we have seen the X, this when this X is substituted or replaced with fluorine group. So, these are fluoro substituent. These benzoyl group, this is the main position of the substituent. These two are ortho, these two are meta, and this one is para. So, when we have a fluoro substituent at para position, these compounds have best antipsychotic activity. If we try to replace this C double bond O group with any isosteric group, isosteric or any functional group, any other functional group, isosteric means those who have similar steric property as that of the CO. Like if we talk about this C double bond O, we can replace this O with NH means we would have C double bond O, we would have C double bond O, C double bond NH. Similarly, we can also replace this oxygen with the sulfur atom. So, we would have C double bond S. So, in this manner, we can replace this C double bond O with either of any isosteric group or any other functional group by changing it like C double bond NH, C double bond S with C double bond CH2. This is also a isosteric compound, CHOH, CHNH2, any other, but that will decrease the activity apart from a single structure, that is Penfluridol. Only Penfluridol is the structure that was found to be active apart from the alteration in the carbonyl group. This is the structure of Penfluridol, in which we have replaced this carbonyl group with an aliphatic, that CH, and further substitution, and still it has antipsychotic potential. So, in this case, diphenyl butyl piperidine derivative, that is CO group, is replaced with parafluorophenyl group. Here what we did, we had here F and we had C double bond O. Now, what we did, we have replaced this C double bond O has been replaced by a phenyl ring with another fluoro atom at its para position. So, diphenyl, now we have diphenyl, two phenyl rings and butyl piperidine derivative. Now, the keto group itself, the keto group, that C double bond O group, has been replaced by a parafluorophenyl ring. Parafluorophenyl ring is used to replace the carbonyl group, and still this drug that possess very good long lasting antipsychotic potential, and including that it does not possess any side effect of sedation, that will not produce sedative effect, and this is the drug that is very useful in the autism. Autism, I would say that is a mental disorder, rather that is a genetic disorder that is being characterized by socially hindered, and especially it has been detected at a very younger age in the children, and so they have a tendency to be socially inactive, or they cannot be much socially interactive as compared to the normal children. So, in those cases it is found to be very useful, because while they are not able to, some of the autistic at the higher range, they are not even able to communicate in a verbal manner. So, in those cases they start behaving like a psychosis patient. So, to treat them, Penfluridol is one of the important drug in case of autism. So, our next target is CH2. So, this CH2 linker group that we have studied, this CH2 linker group, at least it should be 3 carbon atom. It should be 3 carbon atom, either shortening or lengthening or incorporation of any ring that will decrease or sometime complete loss of activity. Like in this example, here it is 1, 2 and 3. Suppose if we use these benzoyl derivative, first the benzoyl derivative, and we increase the chain before the incorporation, before the nitrogen, the difference between the nitrogen and the carbonyl group, it must be 3 carbon atom. Either if we increase its number, or decrease its number, or even by keeping it 3, it is 3 in this case, but by creating a ring, it is now being changed from a normal chain to a ring size. So, if we change this carbon atom into a ring, that will also decrease, and sometimes it is complete loss of activity, means like in this example it possess no antipsychotic activity. So, it must be clear, like the difference between the nitrogen atom and the carbonyl group, the number of carbon atom must be 3. It should not be increased, it should not be decreased, it should not be altered into any ring. In either of the cases, in any of the cases, that will decrease the activity and also abolish the antipsychotic activity of the compounds. Further, amino group modifications. In this case also, like in the case of phenothiazine, a tertiary aliphatic amino nitrogen atom is essential, means this nitrogen atom must be substituted at least by 2 substituents. That here we should have a substituent R1 and R2. It can be methyl, it can be ethyl, but it must be substituted in at the both of the positions, because tertiary aliphatic amino nitrogen, it is essential for antipsychotic agent, and the highest activity is being shown when this nitrogen is being converted into a piperidine ring, and this piperidine ring or the piperazine ring or tetrahydropyridine ring, these all were found to be more active as compared to tertiary aliphatic amino nitrogen, similarly as shown in phenothiazine derivatives. Further, one of the substituent at position number 4 of these cyclic rings, if we produce any substituent at this position and specifically with the aromatic ring, that will also increase the potency and also another benefit of reducing toxicity. So, when we talk about a modification in amino group, the first thing that we have to keep in mind that these amino group must be tertiary in nature. The secondary and primary amino group will have either no activity or they will have lesser activity as compared to tertiary substitution. Further, these drugs will have maximum activity, highest activity when these nitrogen atoms are further altered into a cyclic form such as piperidine, tetrahydropyridine and piperazine. By incorporation of these cyclic forms, highest potency is shown. Apart from this, if we also substitute the fourth position of these cyclic ring, like in this case OH, and this parachlorophenyl ring is this parachlorophenyl acts as a substituent. By substitution of these cyclic ring with an aromatic ring like the parachlorophenyl, that will lead to much better activity, and apart from that, one more benefit, that is also lead to decrease in the toxicity. Further, the different examples of beta amino ketones. When we talk about beta amino ketone, the example is molindone. This is also antipsychotic agent that is very potent and is comparable to the trifluperazine, one of the most potent compound. When we have already studied this trifluperazine in case of phenothiazine derivative. with a CF3, the best substituent, it contains trifluperazine. So, beta amino ketone, the example is molindone, it is comparable to trifluperazine, and it is also used in the treatment of schizophrenia and other psychosis states, like for the decrease in the hallucinations and delusions. So, to decrease those states also we use beta amino ketone, specifically molindones. This is the example of benzamide. Benzamide, the example is sulpiride. Sulpiride is also indicated in the treatment of the schizophrenia, and apart from schizophrenia, it is also used in the treatment of manic attack in the patients. These are some of the miscellaneous agents like olanzapine and oxypertin. These are the examples of the miscellaneous agents. Now, have a glance on the chapter that we have studied different classes: phenothiazine, phenothiazine derivatives, fluorobutyrophenones, beta aminoketones, benzamides and miscellaneous agents. So, first of all, phenothiazine derivatives we have studied. In this case, we have two phenyl rings that has sandwiched one nitrogen and one sulfur, form it in another ring. So, this is the common structure of phenothiazine, in which it is further this nitrogen atom was further substituted, and in this category we have also studied, like phenothiazine, we have different targets. One is this phenothiazine ring, apart from that this alkyl chain and this amino group, and there we have studied the amino group, the side chain, this must be at least 3 carbon atom, the amino group that must be tertiary, and in phenothiazine we have studied only the position number 2 is encouraged for the substitution. Further, we have fluorobutyrophenone, in which the examples are haloperidol and droperidol. In case of beta aminoketones, we have molindone hydrochloride. In case of benzamide, we have sulpiride. In miscellaneous, we have olanzapine and oxypterin. Example of fluorobutyrophenone, these are known as fluorobutyrophenone because we have fluorine at this position, and butyro group, this is 1 and 3 here. So, this is 4 butyro, because it is phenone, because this phenyl group is directly attached with a carbonyl atom. So, this is fluorobutyrophenone, and apart from that you can see the same thing in the droperidol 1, 2, 3 and 4. So, this is again butyro, and there is a phenyl ring with a fluoro and with a carbonyl group. Here again we can see in the case of risperidone, we have found this again the carbon atom, this phenyl, this carbonyl atom, that is being replaced by incorporating a oxazole ring and with a piperazine derivative. This is an example, this is the structural example of risperidone. Further, if we talk about the structure activity relationship of fluorobutyrophenone, so again we have 3 possibilities, 3 targets. One is we can change the benzoyl group, we have already studied when a benzene ring is attached with a C double bond O, then this is known as benzoyl group. So, by modification at benzoyl group, further the modification at CH2 linker group, further modification at amino group. So, we have 3 different targets to study the structure activity relationship of fluorobutyrophenones. First, the modification at benzoyl group. when we talk about modification at benzoyl group, we have first of all the para position, this para position with respect to this carbonyl group. When we have this carbonyl group, so this is the primary substituent, and with respect to this the two positions are ortho, further meta, and the last one is para, where the X is being attached. So, by replacing this X with the fluoro at para position, that will possess the best activity. And by replacing, if we try to replace this carbonyl group with any isosteric group, as we have already mentioned, C double bond NH or C double bond S or C double bond CH2, either of them, that will decrease the activity. Only the compound that have best activity even after the alteration in this benzoyl, the carbonyl of this benzoyl group, is Penfluridol. And so we have studied about Penfluridol also, that in case of Penfluridol we have replaced C double bond O by CH and substitution of para-fluorophenyl ring. So, this keto group is being replaced by para-fluorophenyl group. But after the alteration we have found that it was long lasting, it has a longer duration of action in as antipsychotic agent. Apart from that, it did not possess any sedative effect and very useful in case of autistic patients. Further modification in CH2 linker group. CH2 linker group, we have already mentioned that it must be 3. It must be 3. The number is so strict that even by shortening, even by lengthening or incorporation of any ring in this structure, that will lead to complete loss of activity. Like we have seen the example of incorporation of cyclopropyl ring that contains the same number of carbon atom, 3 carbon atom, still it does not possess any antipsychotic potential. Further, when we talk about amino group modification, as we all know that when we are talking about antipsychotic potential, the nitrogen must be tertiary. Nitrogen in the primary and the secondary nitrogen amino group, that will not have sufficient antipsychotic potential. So, highest activity can be seen when we incorporate some of the cyclic structures like piperidine, tetrahydropyridine or the piperazine. And even more better activity we can see when we have substituted this when we talk about 1, 2, 3 and 4th. This 4th position with an aromatic ring, this substitution also leads to much active compounds. So, when we talk about amino group modification, we must keep, and we are talking about antipsychotic agents, then we must keep this in mind that tertiary aliphatic amino will definitely work, and apart from that, if we can incorporate this nitrogen into a cyclic ring, any cyclic form such as piperidine or the piperazine, that will have much more active compound as compared to the dimethyl, if we say like tertiary aliphatic amino group. And if we want to increase the activity of this piperazine derivative as well, then we must substitute the 4th position of these cyclic rings to have better potency as well as the reduced activity. So, further these are the example of beta amino ketone groups, molindone, and in this category we have seen when we talk about beta amino ketones, these beta amino ketones like this is the carbonyl group. So, this is alpha and this is beta. At this position we have an amino group. So, beta amino ketones, these beta amino groups, they are very potent antipsychotic agents. When we are talking about especially about molindone, it is a very potent antipsychotic agent, and it is almost comparable to piperazine, which is one of the most potent compound of phenothiazine category, and it is also it is used in schizophrenia as for the reducing the frequency and the intensity of other like hallucinations and delusions as well. Further, the examples of Benzamide, the example is sulpiride. Sulpiride is indicated for the treatment of schizophrenia. These are some of example of miscellaneous agents that has been shown earlier. Thank you very much. In the next class, we will study about anticonvulsant agents, and we will get to know what are the convulsions, what are the different type of fits or the epileptic attacks, and how can we manage the epileptic attack. Thank you.